Uh, okay, so welcome everybody. I'm here with my partner, João from Acellea, and myself, Adrià Perez from Computational Science Lab at UPF. And today we'll present PlayMolecule, which is basically a site which gathers several applications that are useful for computational drug discovery, which we'll talk about through the presentation. Okay. So to start, the main topic of our presentation and all the work we do is basically drug discovery, which is a general process that comes from having a disease which there's no cure or you want to improve the cure into a final drug that is released into the market. I know if you have ever seen this, it's very common. It's like a general pipeline that describes the drug discovery process altogether. It's a long process. It goes from 12 to 15 years, and it's a highly cost process, which range into millions, hundreds of millions, and it's too expensive right now. And the main objective of research and all these companies working on it is to try to shorten up the time that you need to develop new drugs and also the cost. And then that comes strongly computational applications and tools to reduce basically experimental costs, which are the most expensive. So basically in the whole process there's two main parts, which are, the first one starts with molecular drug design, where you come out with molecules, and then basically the clinical trials when you actually test them in humans. We'll talk about this first part, which is the molecular drug design, where most of the computational tools are focused, because we cannot simulate humans. So we're just focused on the first part here.
Cannot simulate yet humans.
Yeah. Okay. So mainly in this first part, and also three parts where you can find computational tools that are helpful, are these three first parts. Okay. So first, the process starts with target identification and validation. So you have a disease and you study it and you try to find out which, usually a protein you want to target. Okay, which is it's the part of cell mechanism that you want to either tune up or tune down, you want to turn off, or whatever. So first you identify what you have to do in order to cure this disease. So understand the disease, all right, and basically identify a target protein, which is usually what it's searched for. And you also, when you have this protein, you make structural analysis and you try to find pockets, which these so-called pockets, it's where you will try to find a molecule that can bind into this protein and therefore have a meaningful biological effect in order to stop the disease. Okay, so the main approach is once you have this target, it's to find a ligand to make the effect that you want, and that is basically these two parts, and it's the, I would say it's the difficult part of the process and requires extensive resources, at least for this first part of the drug discovery workflow. So first of all, it's this part here, hit generation and hit to lead. So it's basically you have millions and millions of compounds, and you need to put a threshold and say, okay, and select like probably hundreds of compounds instead of having millions. And then once you have this 100 list of compounds, which are called the hit compounds, then from these hundreds you start searching the best ones, and you come out with lead compounds. Okay, and once you have these lead compounds, you start optimizing these ones that look better in order to then test into animals and then test to humans. You first try to optimize, so when you start doing experiments, you get the good shot, and you don't need to try much, because it's not only expensive, but there's animals there that die, and also if you need to make clinical trials, those are really, really expensive and hard to find. So you don't want to miss your shot. So basically, this is trying to get the best molecule as possible in order to ensure a success in the forthcoming steps. All right. So yeah, I mean, first you start with a high throughput screening or virtual screening with computational tools of millions of compounds, and after you have also one of the main focus in this part is just computing what is called the binding affinity. So you want to compute how strong is the union between this target protein that you're looking for and the molecule you're designing. So you're just looking at that, which is how strongly these two bind in order to create any effect. You just look at this for this part here, and then when you have these strong binders into the protein, then you go to lead optimization, which you look for other properties, which might be also pharmacokinetic properties, which also means how this molecule goes into the parts of interest that you want to focus, or how it is eliminated from your body, which is also important, and well, several other biological things which we're not going to talk about. So yeah, I mean, just because I don't know much the background here, so I'm just going to talk about a little bit about proteins, which is these things that we're focused on finding a molecule. Yeah, okay, so proteins are like machines, like the machinery of cells, and usually when there's a disease or there's some biological problem, medical problem, you can point that into some protein malfunction or downregulation. So what you're trying to search is a way to hack these proteins, so to modulate their effect. And trying to prevent the harmful effects. All right, and one of the most the most important thing is to point out a target protein, and in order to make a drug design based on this protein that you want to target, you need its structure, so its atomic structure. So this is what protein structures look like, and they're all put it up in this database, which is called the Protein Data Bank, or the PDB, which is a basic database for all molecular discovery, or if you any study of proteins, and so on. And that's when that's where the crystal structures, which are called the atomic structures of proteins, are deposited, and you can access them there. And this is all the structures that are available in the world. I think that's it.
Yep. So just to complement here a bit the idea. Also, another important thing here is that sometimes the PDB already contains structures that are of proteins and with ligands bound to it. So that also facilitates sometimes the objective if there's a drug that is already crystallized, already resolved the structure with the binding in the binding pocket, because these techniques of structure determining the structure not only determines the atomic positions of the protein, but also of the ligand, for example, if the ligand is bound to the protein. Yeah. Okay. Yeah. Okay. So what is important here as well, and to move forward in what we are trying to say here, is that people that are trying to do this process of drug discovery, especially the people that are more related with this first part that Adrià was talking about, are normally computational chemists and medicinal chemists. So they are when trying to do some discoveries using computational tools, one of the main challenges is how do these people that are experts in medicinal chemistry interact with these tools. So, because computational tools for drug discovery are not much different from what you see in your laptop or in your computer, right? They are programs that have functionality that is very different from normal computer programs, but the concept is still the same. So something like usability is a big challenge in this regard, because one thing that we saw and working with pharmaceutical companies, which are the companies that are interested in this process in particular, is that a graphical user interface or client, a command line interface, or an API as well, application program interface, these are three ways of interacting with a program or interacting with an end code, with a method that is doing some computational work. But they are very different in the way that people interact with it. And they have advantages and disadvantages. For example, graphical user interface is how most of the people interact with programs, but they are very human dependent, right? You have to click on buttons, you have to click on interact with a graphical user interface. Well, for example, a command line interface, you just need to write some commands, which is much more automatizable, for example, or even more automatizable, an application, an API. So this is one concern, one challenge. Another challenge as well is the setup of these tools. So these tools are sometimes very complex protocols and workflows. And you'll have for sure have listened to some of these tools or have learned about some of these tools in this workshop, and we'll continue to hear about it more for the rest of the days. So normally it's hard as well for individual people or even sometimes IT teams in the companies that are trying to set this up to correctly install and deploy these tools. Because they have particular configurations, they have particular settings. So this is also a challenge to how to set up and make these tools readily available like you do normally for your laptop with, I don't know, a computer game or something else. Then there's also another concern or another challenge, which is infrastructure. So you probably also heard about this, or you're going to hear more about it, which is when you have these tools, sometimes these tools are very demanding in terms of computation. It's not only sufficient to have your own CPU on your computer. Normally you need several hundreds of them or thousands of them. Sometimes you need to have access as well to more specialized computation like accelerators or GPUs. So managing and using computational resources is also an identified challenge for these. So, because there can be a local cluster that is managed by IT team, for example, with some queue managers, for example. It can be a cloud system that are very up to date, right? So it's a new technology as well, or more traditional HPC. So all these things and leveraging all these resources may be also complicated for. Don't forget about who are these people? Are medicinal chemists. They are not the people that want to use this. They are not exactly experts many times on these subjects that we're talking here about. A final concern, a final challenge that I state here is theory and methods. So medicinal chemists, even though they are experts in chemistry, they may not be experts in the methods behind the tools that are being provided to them. Many times they are just the end user. They are not developers of the method. They do not know or it's not their job even to understand fully all the details that were necessary to create that tool. So this is also a final challenge. So here I present what is the main topic of the presentation today, PlayMolecule, which is a way that, so this PlayMolecule was initially developed in UPF, in a computational science laboratory, and right now it's being commercialized by Acellea. And so what is the idea behind PlayMolecule. So the idea is to try to streamline as much as possible all this drug discovery process, all the parts that Adrià was talking about, and leveraging them all together in a single user interface. Many times when you're using computational tools, they're also widespread. They are coming from many companies, they are coming from many different sources. So this is a way as well to try to get together all of this knowledge, all of these state-of-the-art tools, okay, the computational tools, and to make it available to the end user. So one thing that is important here to mention is that these are just tools. The researchers or the people, the medicinal chemists that are doing, they are the ones that need to have a good use of these tools. We're just making it more easily available for people. And as you make it more easily available, it's also more prone to mistakes as well. So it's important to have a human that can use this tool. Doesn't matter how good it is or how robust it is. It's important for it to be used in an appropriate way, because, and we're going to talk about it, there are two main computational methods that we're going to talk about today, which is machine learning methods and MD methods. And it's important for them for the end user, the medicinal chemist, to be able to understand these two methods as well. So one of the strengths of PlayMolecule is its graphical user interface. This is one thing that we noticed, is that we have had some approaches in the past where we can provide to the end users, to the medicinal chemists, very powerful tools, sometimes even automatizable, but many times what the user wants is something that is faster and easy to access. So this is where PlayMolecule graphical user interface comes, where it borrows a bit from the designs of current apps that you see in your mobile phones, or the current web development that is happening, because this is all web-based, okay? So the objective is to have a simple, interactive, in this case, interactive 3D visualization of a molecule, simple menus, not many options, so that people do not get confused as well with too many options, so that things are intuitive, so that things are more easily available for people to use. And this is a screenshot of some of the things that we're going to show today. So, what is available in PlayMolecule in this drug discovery pipeline? So, there are many tools that are already made available. We're going to talk about them today, Adrià and I. So here is just a big general overview over the pipeline. There are some tools here that we're not going to talk about because they are either not available or they're not available publicly. We'll talk about it later. But just to have an overview. So there's many things. Like, for example, there's System Builder, DeepSight, CryptoScout, LeapBox. So all of these applications that you can see here, all of them, they are part of different aspects of this process, okay? You can have heat affinity, for example, lead affinity. These are two different things, as Adrià was mentioning to it. Pocket finder as well. Some more specialized thing like to find allostatic pockets, encrypted pockets. Get binding pose and kinetics when you already have a lead compound as well. And as you can see here, the time to do different tasks is also different from one tool to the other. And here you can see that some of them are based on machine learning, as I was mentioning before, and others are based on molecular dynamics we're going to talk about it soon. So just to give as well a bit of what's happening on the background, because this looks very nice on the front end, right? But what's happening a bit on the background. So you can have here a PlayMolecule, right? You're an end user, right? And you talk. So the PlayMolecule talks to a graphical user interface server that then talks to PMWS. So the PMWS PlayMolecule Web Services is just the back end of that's doing all the work. So this PMWS talks with databases and also runs simulations, so has interaction with HPC, cloud computing through containers. This is important because even though this part we are not showing them that much, because we're actually more interested in showing the application, showing what we can do as well. But in terms of infrastructure, there was a lot of work on the back-end side to make things reproducible, to make things fast to deploy as well. And container technology has been one of the things that has been helping us a lot in this task as well. So yeah, go ahead.
So right now we're going to do an overview of the applications that are available in PlayMolecule. Right, and we'll start with the machine learning based applications. All right, so there's several machine learning based applications, but there's, I would say, more than half of them are based on the same concept, which are deep convolutional neural networks. I don't know if many of you have knowledge in these aspects, but I'll try to keep it simple. All right, these are the applications that are based on this technology, and we'll just overview now. If you can next. So I mean the main idea behind these deep convolutional neural networks is it was to treat proteins as 3D images, basically. So to bring machine vision and convolutional neural networks have been the main development in machine vision. All these ImageNet and basically these algorithms that are able to to recognize elements in pictures and all this stuff. So yeah, we just brought this technology into proteins, and the idea is to treat them, treat the protein structure that is uploaded in the PDB, to treat it as a 3D image. And yeah, basically, so as a 2D image that can be represented in these 3 channels, which can be colors, we can do the same in proteins, but with different atomic properties. If you go to the next one. So yeah, basically we have different descriptions of the atomic properties in the whole protein structure that we divide into different channels, and that helps us in the learning algorithm to reach into relevant relevant information that then it's able to perform predictions correctly. So to speak about these properties, for example, might be hydrophobicity, so how this atom likes to bind into water or not, or aromaticity, which is some chemical property about how strong is a molecule or not, how strongly bound are the atoms in the molecule, more or less like a broad definition, because I don't know if there's any chemical background here or not. But yeah, like different properties like charge or positive ionizable, negative ionizable. Sorry, these kind of properties. What we use to feed up our networks, and to learn, they can be able to predict different things that can be useful in the molecular design. Yeah, I think that's. So yeah, this is an example of how a protein is seen with these algorithms, as I said, treated as a 3D image. So these are the different channels that are put into the network, and then it's able to learn the different properties that we will see in the different applications. So to start with, this was the first application we developed at UPF using this approach. And it's called DeepSight, and basically what it tries to do is predict binding cavities inside proteins. So here we can see an example protein, all right, and what it tries to do is to predict these regions that are like small pockets, small holes, where there's a high probability that, well, it just predicts pockets. So yeah, Yeah, it predicts pockets, which is places that are highly probable for a ligand to bind there and remain there for a significant amount of time, so it can affect the protein. It just tries to find pockets. It's not affinity yet. But yeah, and we trained this deep convolutional neural network, as I explained, with these channels. It was based on SCPDB database, which is a database of binding sites, all right, which is these kind of pockets. And yeah, we later on will show the application in PlayMolecule, but that's the main purpose of this application. Yeah. And then the next one, it's also based on the same approach with these deep convolutional neural networks, but this time is focused on absolute binding affinity prediction. The application is called Kdeep, and basically here it tries to predict how strongly does a ligand bind into a protein and how much. So it is a quantitative prediction. It tries to give this value in this PKD, right, which is basically the equilibrium constant between how much the ligand stays inside the protein or stays out. It's just a measure of how strongly it binds into the protein, all right? And yeah, I mean, it was trained on the PDB bind refine test, right, which is a database for protein and ligand structures only, not just protein and ligand, and it's curated that for only those structures that are really correct and accurate and are useful for these tasks, and which then also has experimental data, so we can train these algorithms based on experimental data. Yeah, you have.
The things that you're feeding the neural network with,
Yeah.
are they just images, or are they like representations of the proteins, or?
Yeah, as I said, these descriptors. So we pick the structure and we basically featurize these structures into these different descriptors. Right? These are like our features of the protein image, which might be the structure, and we feed these features, which are these different channels, and then we said we tell the network to come out with whatever. So DeepSight is give me probability for a binding pocket, and for Kdeep is basically give me the affinity between this ligand and the protein. And in Kdeep, you not only also input the protein, you also input the ligand. We'll talk about that when we show the practical examples, but basically you need the structure and a ligand. When we say what it's called docked, which is a method to put ligands inside pockets, but kind of brute forcing it, all right, with computational methods to move the ligand, but not actual chemistry physically relevant. But yeah, you need the ligand put there inside the structure, and it gives you an affinity prediction, and that's how we train the networks. Yeah, so, and one thing to mention about Kdeep, one thing to mention about Kdeep is that although it's powerful, I mean, here are some targets from the core set of PDB bind. It's another set of proteins inside PDB bind. It has good correlations for most of these targets, but there are targets which has really bad results, and that is because probably there's not enough data in them on the training set, so the algorithm doesn't know much about this kind of protein family. So it's something to keep in mind with these methods that usually, because we have to train them with publicly available data, this is sometimes can be a limitation onto those tools, and it's important to know. Okay, so yeah, I think that's it. And yeah, so talking about that was absolute affinity prediction. So it was trying to generalize over all proteins and ligands to give an affinity prediction, but sometimes that cannot be useful, especially in some parts of the drug discovery process. For example, lead optimization, where you already have a molecule that you know it's a strong candidate to become an interesting molecule to test, all right? But you need to better improve its affinity. Which is called lead optimization. So usually what you do is you have this lead compound, and you generate similar molecules that are not, these are molecules that are not very far structurally speaking, Okay, so they're just small modifications, maybe in some part of the molecule or another. It might be based on some structural information or not. I mean, usually in medicinal chemistry, what they do is they perform experiments and they look at the structure and the molecule inside the pocket and try to think what modification might help. Okay, but this in experimental, I mean, if you want to test this experimentally, it takes some time and some resources. And the baseline methods to do this with computational tools, it's called free energy perturbation, which is an MD-based method, but it's also very heavily, it requires high computational resources, and it might not be available to everybody. Also, it's has some. We thought on it. So we thought on developing a method specifically for this task, which is lead optimization, which basically tries to make relative binding affinity predictions. So instead of predicting the absolute affinity between a protein and a ligand, we try to predict the difference between affinities. So if you know that this compound binds with some defined affinity, then what you try to predict is how different will this ligand be based that I've put this atom here. So will it be will it strongly bind into more strongly bind than this one, or will it be worse? So it just predicts the relative affinity. So next slide. So basically, the concept is similar to Kdeep. It basically predicts affinity, but what we use is zero symmetrical neural networks. And basically, what we try to predict, here we compute ligands for separate, and then we make the difference here. When you put them together, you actually make a difference between these latent representations, and then you get the output is the difference between the affinity of one ligand and the other. So maybe this one ligand had 10 in affinity and the other has 11. So what you're trying to predict is this difference, which is 1. So you don't need to have information on the whole thing, but just yeah. And here is an example comparing some so comparing some congeneric series, which is the series of similar molecules, using, you cannot really see it here. This light blue is absolute affinity prediction, so Kdeep, and here it's relative affinity prediction. So it really increases performance compared to an absolute affinity prediction, which is, I mean, they're just different tools for different parts of the drug discovery pipeline. All right, and next slide. Here, there's just some results compared to FEP, which is like the baseline for this prediction problem. All right, and here we're comparing to this benchmarking on this Schrödinger dataset, which is basically the baseline on FEP that we had to break. And the interesting thing here to see is not that we show similar performance in terms of prediction, which much less computational resources. It's not the same to perform simulations that to execute something for less than a second. But also interesting to note here is that because to perform this, what you need to do is you need to retrain the network with your congeneric series. So you add, for example, 3-4 ligands into this training set, and you can keep continuing to add new ligands as you make, for example, experimental predictions of binding affinity. And if you introduce them inside this network, you'll see that for several targets, the accuracy increases as you add more ligands into the knowledge of the network. Okay. Well, just going back a little bit to a previous step in the drug discovery pipeline, we also have BindScope, which what it tries to do, basically, it has a broader scope even than Kdeep, where basically this is just focused on making active-inactive classification of ligands. It's also based on on the same concept as Kdeep or DeepSight with deep convolutional neural networks. But here it's only based, it's only trying to differentiate if a ligand is going to bind into that protein or not, which is a different classification, different problem from computing the affinity. And yeah, here I just wanted to show that it suffers from the same problem as Kdeep. So for protein families that it has seen in the training data set, you have like really good predictions, but maybe for other families that you don't have that much information in public databases, then your prediction is not going to be that good. BindScope was trained on DoD database, which is a database of proteins and docked ligands, and it also has information on ligands that do not bind into proteins. But it doesn't have affinity predictions, affinity data, so, and it's much more extensive than PDB binds, so that's why the database is different. Okay. And then we have LeapBox. This is a little different from the other ones. It doesn't quite make, well, it does make a prediction, but it's based on a different concept. Here, I mean, it still uses the deep convolutional neural network concept, but instead it's treated as a, well, it's converted into a generative model, so it tries to generate data. And basically what it does is you give a structure and you define the pocket area on this protein, and what it tries to do is to generate these ligand properties based on the pocket information. All right, so it tries to so when you look inside the pocket, protein pocket, it tries to give information on where you should have properties in your ligand inside that pocket. So when you look inside a pocket, maybe this model will tell you, okay, here you probably need aromatic atoms, and then here probably you need a nitrogen donor, and here you need a nitrogen acceptor, and these different properties which devise inside this pocket. Joe, next slide, please. And here is an example. Okay, so you have this pocket here inside the protein, and it tells you, okay, all of these should be aromatic, and here maybe you need bond acceptor, hydrogen bond acceptors, and here you need hydrogen bond donors, and so on. We'll see that later when we'll show you the tools, and you'll probably see it more clear. And then we also have another generative model, but this case it's for molecules, and it's called LeadRing. And yeah, basically it's a shape-based generative modeling, which tries to, when you input a seed molecule, which might be a lead compound or a hit compound, and what it does, it comes out with similar molecules, which maybe you haven't accounted for. So usually a medical chemistry that is trying to develop new molecules, which already has a lead compound, which might be this, he has some ideas on what he can do, but maybe he's biased because he's a human and has more experience. I mean, he has these human feelings on that, on this modification might work or this might not, but maybe he's not accounting for other modifications that are possible. And basically this tool, what it does, it generates ideas that might give inspiration in order to develop useful molecules. And yeah, basically the concept is based on, you give us a seed molecule, which is the starter molecule, and you use a shape autoencoder, which is based on convolutional neural networks, in order to make a shape representation similar to what we had with LeapBox. And then with this shape, basically you use this shape captioning, which then later gives outputs molecules. This is the basic idea, and this, well, I'll show you now next. This is like the more technical representation. So you have the compound representation, which you encode with 3D convolutions, and you have this latent space that encodes the shape, and then you decode this to generate new shapes based on this latent representation. You can also input here compound pharmacophores or other information, and then once you have these generated shape representations based on this encoded space, you have the shape captioning, which is basically a tool to go from these shapes into smiles, which is a way to represent molecules with text, and from these smiles you just get the molecule. Okay, I'm not gonna go very much into these technical details, but here is next. Here's an example of of molecules generated with this tool. I mean, problem here is that it's not. I mean, it's hard to evaluate how good is this because, well, in order to evaluate, you will just need to test these molecules and see if they are effective. The idea is that it generates molecules that are probable or highly likely to bind into the pocket because the idea is that it gives similar information. But I mean, it's harder to evaluate how good is it. But I mean, the most you can say is that they look well. They're logical with a chemical point of view, and that's the most thing we can say here. So and then as the last machine learning based application, it's pathway map. Which basically it's as it says there, molecular pathway predictor. So this is when you have a ligand, it might be lead compound or whatever, or for example, there's these several web pages when you can buy compounds. You might be interested to know if this ligand is going to bind to other proteins besides your protein. Because it's something that usually happens. So ligands are not specific for one protein target, but they might also affect other proteins, which might affect other pathways inside your body, which might be, so maybe you're targeting a specific type of cancer, I don't know, like lung cancer, but then this ligand that you designed for lung cancer, the specific type of lung cancer can also affect your brain or can also affect other pathways. So basically what this tool does, it tries to predict where a ligand, which pathways a ligand is going to affect based on different assay information that has been. And it's also useful in order to identify active compounds within dark chemical matter. This dark chemical matter, it's basically these compounds that have been designed, but there's no protein, there's no known proteins where this ligand binds. And it might be useful to identify and give usefulness to this dark chemical matter, and also for the orphaning compounds, which there's no effect known on it. We'll show after that, and you'll see more clear how it works. That's it for the machine learning based, and we'll continue with molecular dynamics.
Yeah. So Adrià talked now quite a bit about machine learning applications, and now I'm going to talk a bit about molecular dynamics applications. And to start that, I will start by telling you what is molecular dynamics. So molecular dynamics is a computational method that allows you to simulate a system at the atomic level. So we talked before about protein structures, so atoms in space, but they are just like an image, right? Just a still image. But we know that atoms are not still. They are moving, they are dynamic. So this tool tries to, through an atomic model of the system of interest, in this case that we're talking about protein ligand binding, we try to observe or in this case simulate or observe through simulation of this mechanical behavior of the system. So what is the protein dynamics? How does a ligand reach the binding site? How all of these dynamical properties occur in our system. So one important thing here to give you a general picture is that we have here the size of the system, so the size of what you're trying to study, and you have here the time scale of what you're trying to study, and you have here a conjunction of biophysical techniques that are used experimentally to study atomic behavior, atomic dynamics, or atomic structure. So you have here NMR, you have electron microscopy, electrophysiology, you have FRET, AFM, X-ray crystallography as well. So most of the structures that we see in the protein data bank come from X-ray and NMR. Okay, so these have very limited scopes of size and also of so the scale and also of time scale as well. So size scale and time scale. Here you have molecular dynamic simulations, which it has a broad range of sizes that it can reach, but it still doesn't reach the time scales that sometimes is important for some of the processes that you're interested in. For example, protein ligand binding, normally it can span all of this time scale, but some effects or some systems actually they are on slower time scales, okay, and we haven't quite reached that yet through molecular dynamics. But it's as computational power and methodology moves forward and progresses, this barrier gets crossed more and more. So maybe 10 years ago we are right here, right? And 10 years after we are here, and 10 years after we continue, and then we'll reach seconds time scale, which is could already be sufficient to observe many of these processes. So how does exactly these simulations occur? So we need a physical model of our proteins, or we need a physical description of how does this dynamics or this mechanical model, how does it work. So basically this is done through molecular mechanics, through force fields. So this is basically an equation description of the energy function that describes the system. So basically we have different types of atomic interactions that we describe with this system, with these force fields, which are bonded or non-bonded. So this is just a model, an atomic model, a physical atomic model that we have for our system of interest. So you have bonds, you have angles. For example, you have bonds. A bond is a connection between two atoms. So you have basically an harmonic that describes how does this bond vibrate. Or you have, for example, angles as well, which you have three atoms, and you see how this angle vary, as well as, for example, torsional angles, which are for four atoms. So how do four atoms, how do these two planes formed by three atoms, how do they, what's the angle between them? So all of these have force constants that are correspond to them, and then you have non-bonded as well, which means normally two types of interactions. You have van der Waals, and you also have Coulomb interactions as well. So all of these sum together, and summing as well throughout all angles, all bonds, and all torsions, so on and so forth of the system, you can have a complete physical description of your system. This is what's called the energy function. Okay, and then the parameters that we talk about are these parameters of the function is what constitutes the force field. One important thing here to understand this is that normally we're talking about systems that can be 30,000 atoms, 50,000 atoms, 100,000 atoms. So normally these parameters can escalate very quickly, and it leads to computations that are quite demanding. Okay, so now we have this model. What do we do with it? So what is actually molecular dynamics? So basically we have this model, and we apply an integration algorithm, which is based basically on the Newton equations of motion. So you have the initial positions of the system. So We have the model, yet we have as well the protein system with a ligand and water, okay? And we have its initial positions, which is taken from the structure that we already have, okay? And from the preparation. We'll talk more about that later. So then we choose a small time step, and then from our initial system and this energy function that we have, so our model of the system, which is a physical model, we can get the forces by deriving having the gradient of this energy function. And with the forces, we can calculate what's the next position of the in time, the next position in time of this system, okay? So then we just update the initial positions to the new positions, move the time forward, and then we iterate. So with these new positions that we just calculated, we calculated again under our model the new forces and so on and so forth. And we do it iteratively for how much time we want. Then it depends on the computation that we have available. One important thing here is that the integration step. The integration step, which is this short time step, normally to be meaningful, and this is one of the major limitations, it's in the orders of femtoseconds, okay? This is the question of the time scale that I was talking before. So femtosecond is 10 to the minus 15 seconds. This is because we cannot do a simulation where we just jump 1 second. It's not possible. It needs to be a small jump for all this thing to work, okay? For the integration to work. Yeah, so then there's a lot of focus on trying to optimize this. So this is a very simplified view of what is MD, okay? Then there are very complex algorithms with different types of integration and so on and so forth that can be optimized, okay? So that it goes faster, it computes faster, and so on and so forth. So here, if internet allows us,
no.
Does it?
Yes.
Does it allow it?
It
Yes?
allows.
Okay, just let's see how internet is going to help us here or not. So basically, the idea here was to try to show you a video of molecular dynamic simulation, so that you could understand it a bit. Unfortunately, it's kind of.
Wow.
Can you stop it and try to see if it buffers better or?
Oh, yeah. Maybe.
Yeah, because it doesn't have a good connection, so it changes resolution, I think.
Okay, let's try now. Yeah, well.
Okay, I'm sorry for the bad resolution. Well, it's improving. So here you have a protein that you can represent here in white. Okay, it's represented this white, and you can see its dynamics. Okay, protein is moving. Okay, and then here in blue, and then also in red, you have a particular ligand that is basically exploring the space around the protein. So what we can see here, this is a result of many simulations. Okay, in red is a successful binding process. Okay, so actually here we know that is the binding site, and one of these trajectories actually reached the binding site, and the ligand actually fit there. So this is it's here some information about the data. So yeah, you put it again, yeah. So here, for example, you have another ligand, this one in blue, that here it's exploring, but it doesn't actually reach the binding site. So these things happen, and it's actually part of the process itself, because the affinity that Adrià was talking before, how strongly does it bind, it also depends on how strongly does it go to the binding site, or how can it explore this space, okay? So what's its affinity there, and also what's the kinetics of getting to the binding site as well, okay? So this is also why it makes molecular dynamics such an attractive tool, despite being a model and being a simulation. It's such an attractive tool because most of the tools that are available experimentally to see these are much more limited in a way that we cannot observe as we are observing here. That allows us to derive some mechanistic view of the process. So here, I'm trying to focus from general MD, so MD can be applied to many things, okay? material chemistry. I don't know. There's the field of molecular dynamics. It's very wide. But trying here to focus, and we're trying here to focus on the subject that we are talking about here today, which is drug design. And we can design. We can have an MD workflow for this, what we call in silico binding assay. So basically, what we showed you before was a protein in water. The water was excluded for visual purposes, because otherwise you wouldn't see anything, because it would be basically a box full of water that you wouldn't see anything. So this ligand starting from a position outside the protein, and when we don't, in this case we knew the binding site, okay, but when we don't know, we allow the ligand to explore, we allow this small molecule to explore the space and to reach and find its binding state, this binding spot. This is what we call a binding assay, so the in silico binding assay. So there's binding assays that you can do experimentally. You have a test tube, right? You have your protein there, and so on and so forth. You put there some ligand, and then you will measure some experimentally some activity or something like that. So this basically draws from the same concept. But to do this in silico binding assay, we need to create an MD workflow. So this seems very simple as I'm explaining it to you, but there are several steps, since you have, from the point that you have a protein structure until you actually perform and do a molecular dynamics simulation, that essentially are resumed here, okay? So you have an initial preparation, which means that you need to prepare your protein, build a membrane if it's a membrane protein. You have to parameterize the ligand, because this is a small detail, but basically when we have all this chemical space that is available, all these parameters that I talked about before, they may not be available, and you may need to get them, because they are only made available for the most common compounds. For example, for proteins, the parameters are available because everyone simulates proteins. But if you want to simulate a particular compound that you have just discovered, a new lead, a new hit target, so it probably will need to parameterize it. Then after you do this, you need to build your system in a way. You need to solvate it, so you put it in a box of water. You need to ionize it, because we are not living in pure water, okay? We have ions, and it's also important for charge balancing. Then you need also to place a ligand in your system as well correctly. You need to then assign the parameters. So you have all of this structure. So you have all of your system. Now you need to assign the parameters and be correctly assigned, so that you have a working simulation, okay? Then after all this process, you actually need to run molecular dynamics simulations. For that, you need to do an initial minimization, so that it releases some of its initial strain. Some of these systems, the structures were determined at a temperature that is not the room temperature, okay? So the system needs to heat up a bit, so it needs to minimize some stress that it may have. Because of the same reason, we also need to calibrate it as well, to bring it back to a kind of equilibrium state. And then you need to do production after you read this equilibration. Then it's possible to have some more complex responding schemes, where you have some sampling schemes other than using molecular dynamics, but you have a bit more, they are smarter in a way. And then after you run this, basically what is the end? It's what we're seeing in the video, right? You see some points in space, and you have what we call a trajectory. Basically, it's a film of what's happening in the protein, okay? What's happening in this system, okay? Then, how do we get information from this? So you can visualize, like we were saying, okay, this ligand goes to the binding site, okay? Or you can have some more complex analysis tools like Markov state models. You can calculate properties, and you can also visualize trajectories. But there are many ways of analyzing and seeing this. Okay? Is everything clear until now, or do you have any questions? No? Okay. So now I'm going to talk about what is available. Sorry, can you go back one? Yeah. So this is similar to what we were doing in machine learning. So all of these different parts, there are different tools that we can use to help the medicinal chemist to do all this part without having much effort, okay? Because that's the part I was trying to convey here, the idea is that all of this is a lot of work unless you have a tool that allows you to make it much easier and faster and automated. Okay, so one of the things that we're talking, one of the tools that we are, is protein prepare. So this protein preparation for MD simulation. So what is protein prepare? So proteins need to be prepared at a given pH. So normally, this is one of the details that is important. So pH is an important state. It's an important property that we have. So normally we have proteins at pH 7, or we have our system at pH 7. Sometimes it differs on the different parts of the body. So, but this model that we use, molecular dynamics model, they don't allow change in protonation states, which is related with pH, okay? So normally, we need to fix a protonation state in the beginning of the simulation and carry on with it. This is the normal behavior that we expect. There are more sophisticated methods, but we're not talking about them today. So normally, we need to actually know what are the protonation states of the system, of the protein at a particular pH. So protein prepare helps you with that, so that you don't need to actually choose the protonation states by yourself. So basically, there's here some pipeline that we use to calculate and assign the pKa values of each protonable residue that exists in the protein, and you optimize the hydrogen bonds as well. And this tool, Protein Prepare, allows us to do and to look at how was the system prepared as well by visualizing it. You can also see the different residues that are in the protein, the different titrable residues, the different residues that are protonable, what is their pKa, so what's their affinity to protons or not, okay? And this allows you to make decisions, or allows the medicinal chemist to make decisions and to choose a system better, to choose better the system for carrying on the rest of the pipeline. Then we also developed a tool called Parameterize that allows you to get these parameters that I was talking about. So if you have this huge chemical space with all the molecules available to you, so you need to have a way of obtaining these parameters to perform molecular dynamics simulation. So this tool developed basically it consists of 3 steps where we first start with some basic parameters that we have, and then we can parameterize most of the we can parameterize some of the most interesting parts, which are how does the dihedrals, because this is one of the part that is more problematic, which is dihedral angles. They are poorly parameterized in by default, and then we can do that by doing calculations. So we can do this by doing expensive QM calculations. QM is quantum mechanical calculations. Okay, so going to more expensive computational methods than molecular dynamics, but more accurate. Okay, to calculate the energy. But actually, what we did here in Parameterize was to train a neural network potential to behave like a quantum mechanical calculation, okay? So that it learns from quantum mechanical data, and then it can predict quantum mechanical energies, okay? And then when we have this parameterization, we can, after we do these calculations, we do these predictions of rotating the bonds, and we can actually fit our potentials, fit our parameters to those results. We have here some examples. For example, this is one general plot where we show that the initial parameters that are there available, that are made available for everyone, we can dramatically improve them in many cases, okay, for some general data set or for some particular drugs that are already existing in the market. These are 4 drugs that exist in the market. One example here, if I'm not mistaken, this molecule is this one. And we have here a particular, so we have 2 dihedrals. We have this one, which is this one in particular. So you can see that the initial parameters that were here, okay, and the green line here is the target parameters that we actually want, the realistic ones, okay. And we see that our parameterize tool can reach those, or can reach very close to those parameters, okay. So another tool that is important as well, so we have these two initial preparation tools, then we have the building tool, we have this system builder. So after we have all the components, okay, the ligand, the protein, the membrane, we can merge them together. So this is a complex tool that allows you to insert the protein in the membrane if there's a membrane, okay. You place the ligand in the solvent area, you solvate the system, you ionize the system, and you assign parameters. So all this process is done by this system builder that is an automated tool. This used to be lots of work, but right now we provide a tool that is much more easy to use, and it's much more interactive for the medicinal chemist. So here, actually those were inverted, it doesn't matter. Okay, so this is another important part here is that many proteins that are relevant for biological functions, and for example GPCRs, which are the main target of pharmaceuticals that are available in the market right now, they are membrane proteins. Okay, so other than just modeling the protein itself, we also need to model the membrane and insert the protein inside the membrane, okay? And then for that, we need to build the actual membranes for the molecular dynamics, as they are not readily available like protein structures, okay? There's also an important detail is that membranes can also have complex compositions with different lipids, okay? And different percentage of lipids in different layers, okay, of the membrane. Then I'm going to talk to you about this, but we're not going to see it today because we're going to show you the public version of PlayMolecule. And since molecular dynamic simulations are a bit expensive, they are not made available publicly, okay? Because this server, the playmolecule.com.org, it allows for anyone to use it, but it doesn't have infinite resources, okay? So we limit it in a way. So this tool is not made available right now. It may be in the future, we'll see as well. So basically, we have two different tools of doing molecular dynamics. I'm not going too much into the details here. So basically, we can do a simple run, which is basically we do a minimization, equilibration, production, which is exemplified here with the schematics of how does it behave, okay? And we can also do adaptive sampling, which is an improved sampling improved sampling of important conformations using Markov state models. So I'm not as well going here into the theory, but basically it's a learning MD, okay, where in different epochs, okay. So instead of running one molecular dynamic simulation, we run a bit of molecular dynamic simulations. We do a model of our system, a Markov state model of our system. How does it behave? Okay, what are the kinetics of our system? And from what we learned there, we respawn new conformations of the ligand, new ideas, so that the molecular dynamic simulations can start from there. And this continues for the next epoch, where we do more simulations and we find new states and we continue on and so on and so forth. And what has been shown is that this improves the sampling of the system, and you can reach much faster the binding site, okay? Then and also improve as well the energy, the affinity of the compound, the prediction of the affinity. And finally, also one thing that this allows is to manage resources as well for the end user. So the user just goes there and says, oh, a simple run, I want this and that, I want to run this system that I have here, I want to do this amount of nanoseconds of simulation, and then the user doesn't need to worry about what are the resources that he wants to use, what are the commands to submit to his cluster. He doesn't need to think about that because the system will do it for him. Okay, PlayMolecule will do it for him. And now just one final thing, which is still in beta. It's a new thing that we added to PlayMolecule. It could be interesting to just show it to you as well, which is this Plex view. So when we have this protein and ligand, sometimes it's not clear for you to see how they interact. So this is a good tool for visualization of protein-ligand interactions in a clear way, where you mark more important interactions like hydrogen bonds, pi-pi stack, and so on and so forth in a visual way, in a 2D way that sometimes may not be as obvious from a 3D visualization. And the objective is as well to integrate it in the near future with the MD trajectorial analysis. And a final piece of PlayMolecule is just databases for computational chemistry. So one thing that we noticed as well is that PlayMolecule has the potential, as well as many other websites that have existed in the past, to be an easy way, an easy platform, an easy interface to interact with databases as well, okay? So one thing that we did was we did these simulations to test actually our tools of many systems of proteins in membranes, okay? Which is actually from a database which is called OPM, which is a database for eukaryotic membrane systems, okay? And we made these simulations, okay? We made this paper, and then we made all trajectories available through PlayMolecule, okay? So this is a way that, since MD trajectories are so expensive to calculate, this could be a good idea as well, to make trajectories available for everyone. So as a database of the MD trajectories for easy visualization and analysis. So this is a snapshot, an example. So I'm not actually going to show this because we may not have time, but basically where you can visualize the trajectory, you can play it, you know, you can see some properties being plotted as well, okay? And also the possibility of downloading these trajectories yourself, you don't have to calculate them, and then analyzing them locally with other tools that you may have as well. Another thing that is also interesting is that we created recently, which is the PlayMolecule data center. So one thing that is important as well is data validation, okay? And one thing that PlayMolecule were having some problems as well was how do we deal with some databases that we are, for example, imagine some of the tools that Adrià was talking about, and some people want to submit a library of compounds to do some kind of affinity prediction, for example. What about what you do with a 50 million database, 50 million compounds database, okay? So this better management of data files was a necessity for us because of the size and also the quantity. If you have many files as well to do. So we developed this data center, which is also interesting as well, where you can immediately upload your data, it gets validated, and then as soon as it gets uploaded to the system, you don't need to re-upload it again from every time that you want to use it in an application. You just reuse it from the data center that is available. This is also interesting as well for application integration. So you have different applications and they produce different outputs, that the outputs of one application may be the inputs of the other. So this allows you to carry on from, we don't have to download the results of one application and then re-upload it to the next one. You can do this direct management, which makes integration as well much more streamlined. And to finish up a bit, and since we are here, CompBiomed is organizing this workshop, so one important thing as well for us in this consortium as well is to try to, since we have this very nice interface, it's to try to make some of the tools that you probably heard about it today and you're going to hear more about in the following days, make some of these tools that are available to the consortium, okay? CT2X, Bond DVC, Visual GEC, BAC, antibiotic resistance. So all these tools, they are possible candidates as well to be made available in this platform that is much easier to use than some other tools, okay? And until now, PlayMolecule, as the name suggests, it has been basically on molecular medicine, okay? But in the future, this may be expanded as well to other fields of drug discovery, because we've been focusing about the molecular discovery process, but there are other things that are also important, that, as Adrià was mentioning as well, like what is the effect of the on the blood flow, on the bones, so there are other parts, more macroscopic parts of the body that you can also do some modeling, you can also do some visualization as well, and it would be interesting as well to have it in PlayMolecule. So, and with this, I think we just need to finish by, oh, this is so weird. Okay, by acknowledging the people that are actively working on this, or from Acellea and Computational Science Lab. So, Adrià supervisor and my boss, Gianni Di Fabritis. We also have the PlayMolecule team, main PlayMolecule team that is here, Gerard, Alberto, and David. We also have many people contributing with applications, machine learning researchers, molecular dynamics simulation researchers, ComCam, so on and so forth, drug discovery specialist, Davide, we have Micha, Jose, Raimonda, Stefan, and Alejandro. We also have to thank the funding as well, and I think it's all, right?
That's all.
And and it's if you want to contact us for any reason.
Now, well,
wait. So there's already questions. I like it.
It's just for curiosity.
No.
I understand that TextView version of this or whatever is able to predict the interaction between residues of several proteins. And talking about maybe GPCRs and the possible different interactions of each drug, which potentially activates the GPCR. Would this program be able to determine which compound triggers certain protein modification? Because we know there's biosignaling and then beta-arrestin and GQ signaling. Would this kind of predict if a new molecule or a potential drug activate one or another, or even get a dual one, or maybe, I don't know, a time-depending dual activation GQ beta-arrestin? Would this help us?
Okay, so you're talking about PlexView, right? We're talking about, but PlexView is just visualization, the final visualization. What I could suggest to you is the entire pipeline, of course. So we have actually one tool that I didn't talk about here as well. We try to predict as well a lustery, which is this type of effects as well. You can also try to make your own models as well, where you put one. So we're talking about GPCRs, where you have the two different modulators as well, right? You have to do different binding sites. So here you can do some experiments where you can do some MD with one of the binding sites within one compound, and you try to do a binding assay of one of the others. Okay. You can also, using the machine learning tools, you can also do the same trick as well, where you pass a protein that already has a ligand, okay, there, and see if there are some differences as well. But so for the machine learning tools, we're talking about more high throughput. We're talking about more getting numbers, okay, getting predictions, and not about, I think, which you were more interested, at least I understood, which is more maybe the details or maybe the mechanistic view. For these things, of course, molecular dynamics is much more interesting because it allows you really to see the process and see the kinetics. Of course, it also means that it's much more expensive.
All right, so now we finished our presentation, but we'd like to show you PlayMolecule itself and how it works. All right? All right, and here are all the applications we've been talking about, okay, and even more.
Yeah, we didn't plan much this, so okay, so this is going to be a bit informal, okay? We're just going through some of the applications. Adrià is going to talk about some, I'm going to talk about some, and we go. I mean, it's also, if you want to have some interaction, you have some doubts, just ask us, okay? So it's more or less hands-on, so you can do this by yourself, okay? Because you have available the website, you can click on the applications, you can try it by yourself. They are very intuitive many times, okay? So, but we are going to showcase. This is more of a showcase, okay?
All right, so, well, I don't know if there's a particular interest in any application that we might look into it. But if not, we'll just, for example, start with the beginning, all right? So imagine you have a protein target, and you like to study it, and first of all you start making a structural analysis and trying to find, is there any pocket that I can target my ligand design on based on this protein. So you will need to predict which pocket you want to target, right? So first of all you would start with DeepSight, okay? So wait, I think this is already the result. So here is DeepSight. All right, let's see if the internet helps. So this is the interface with DeepSight. All right, here's the input part. That is all of this is common with all the applications. You'll see that, and every application has different input necessities. Here with DeepSight, we just need a PDB ID of the protein structure. So basically, the atomic structure of protein. You can either put yourself your own file of your own generated PDBs, or you can just put the PDB ID from the PDB database, and it will automatically download it. So, for example, we'll start with this example. Okay, you select. There's usually different chains in proteins sometimes, so you just submit it. Okay. And yeah, I don't know how it will last. Yeah, I think this is the same because I just uploaded. Well, it's already done. So
yeah, it's
fast.
Quite quick.
Yeah, it's fast. All right. So no, it's still computing. Here. So it's the same protein as before. So here is the prediction. Okay, this is the graphical output. You can see with this surface where the pockets are predicted to be. Okay, but then you have like more numerical results. Here are the top ranked pockets in this protein. Okay, with the different scores. Here are the coordinates, and you can. Well, that's too much of a zoom, but it points out to you into this. You can toggle the surface, and also I think you can
toggle it
make it more transparent.
Yeah, it should be removing it.
Yeah, right. I know.
Yeah.
But yeah, basically you can move out the surface to see with more precision or not where the most probable pockets are. And if we okay, so we have these targets here, and you can just look at it, and you can you can look at its surroundings if this was working correctly. Probably it's because of the internet. It's not being that much interactive, but with good connection, it works. And yeah, basically it ranks the most probable pockets on your prediction, and you can also I don't know why is that much. You can also download the results you've got with the prediction, so you can. There's also more pockets inside this text, and you can just parse it into or use it as you want. So once you have this pocket, maybe you want to learn more about it and see, okay, I'm putting this pocket in here. I want to know more about the properties that will be good in order to target this particular pocket. So we can use LeapBox, which I have here. Okay, it's the same example as before, and you can see here that this is a selected pocket. Yeah, so maybe we'll start by inputting the file and see how this works, because this has so yeah, so you upload the protein. Okay, this is 3PTP, and to input LeapBox, you just have to give an area that you want to study. So usually that would be the pocket. You can select the box center where you want to be. You can move it around. Okay, we'll just focus it on where an actual ligand is in this structure. So it's an actual pocket. Okay, and the result is this one here. All right, so it gives you more or less an idea on how you might want to fill up this pocket in order to have a strong binder there. So to have a good ligand candidate. Okay, and here you can look at this field that the generative model outputs to see where you would like to have aromatic atoms, or for example hydrogen bond or accept. Ooh, happened.
Just
yeah, what would you like to have hydrogen bond acceptors or hydrogen bond donors, and you can see different thresholds on the information. You can also download results with more numerical values and so on with the surface.
If you go back to the other example, you can see
Which
that,
one?
no, to the input, where you put the input.
Oh, yeah, here.
So you see that the prediction is actually quite good, because the ligand that's here, it's benzamidine. It's basically an aromatic, and it has this donor. So basically it predicts, in this case, by the shape of the pocket. Don't forget that the one we passed actually a structure that has a ligand, okay? But this ignores the ligand. Okay, this is just doing that prediction based on the protein itself, okay? Because we actually know that this protein has this. This is just an illustrative example. Normally we would be passing a protein without a ligand, okay? So this shape, it's predicting, right? Like Adrià was saying, the aromatic part, it's this part, and then the donor part is this part. So it's actually doing what we were expecting it to do, okay? So if we were to design a ligand to actually fit that pocket or fit that space, you would have more or less a good prediction of it.
So yeah, this is a tool that gives you structural information, but more focused on designing a ligand on the structure of the protein. And okay, so then I know, for example, you want to yeah, maybe you want to test some ligands in this protein. Well, actually, I have another example with more. This is my example. Okay, so let's start with Kdeep. I'm gonna close this. Okay, so with Kdeep, what you need to make it work, it's just a protein. Basically, you need a protein structure, as most of them, and then you need docked ligands. So basically, you need information about the ligand structure, and also you need them to already be put inside the protein with other computational tools, which is mainly docking, which mainly puts the ligand by moving it and making several different algorithms and ranking the positions, but you basically dock the ligand, so you put the ligand inside the pocket in what might be more or less the binding conformation, okay? But it's very fast computationally, but usually not very exact, so you usually make lots of docked structures, okay? And you just basically, yeah, you need the protein and the ligand docked inside in the protein pocket, and then you pass the input into this algorithm, and it gives you an absolute binding affinity prediction. Okay, so yeah, if you input with your own PDB and the molecules you have available, and you want to test the affinity, and here I have an example with 4 molecules. All right, and here's the graphical output of it. So you basically have the ligands that you have put in the SDF file, and you have molecular weight, and here is the prediction. So actually Kdeep, what it does, it predicts PKD, which is this equilibrium constant, and from that you can also output you can also compute the energy value of this binding, the free energy, which is in these units, the free energy, yeah, in kcal mol. Okay. Ligand efficiency, I'm not sure what that is, but well, yeah.
The ligand efficiency is just you divide the. So basically, what ligand efficiency sometimes is interesting for medicinal chemists is that molecules that are bigger, okay, that have more atoms in general, they're going to be better binders because there are more interactions that are. So ligand efficiency divides the binding energy by the number of atoms that exist in the molecule, okay? So basically it gives you like what's the binding strength per atom, basically, okay? More or less. And sometimes it's used as a rule of thumb by medicinal chemists to try to see actually if a compound is strongly binder or not.
I mean, it gives you information on how strong your your ligand binds into the protein and gives you useful information for the medicinal chemists to later make decisions. All right. Yeah, basically that's Kdeep. And maybe from Kdeep you already have information. You select a lead compound, okay, and you want to generate similar compounds to that one. So let's go to LeadRing and try to make new compounds and generate new ideas. So here you may already have your own molecules that you want to generate more, but maybe you can even try with this drawing in order to test. So we can just draw a molecule now. Okay, we'll start with this benzene. Let's see.
You should have clicked on the bond instead.
Ah, okay.
But
All
doesn't
right.
matter, doesn't matter.
No, it doesn't matter. So, all right, so here, right?
Yeah, exactly. Yeah.
Okay, so we can start with these. Maybe I can put,
See, it's intuitive, but you need to.
I
you need
can maybe, no, I don't want this here.
So the objective here is to try to, I think the idea that we're trying to pass here as well is trying to, these things that we are showing here, normally they are made through separate programs, separate interfaces. So we try to put in a single platform all the tools that are necessary, which makes it much more attractive for the end user.
Okay, so
go
we
ahead.
have this example molecule and we just submit it.
See?
Oh, okay, I did an invalid molecule. All right. It's probably this.
You wanted to change it to,
Yeah.
I don't think it's,
Oh, yeah, wait. Oh, it's here because, yeah, this is wrong.
It's, yeah, put single bond. Yeah,
All right,
that should be okay.
so we submit it.
So, and you can also do these things yourself. So if you have, you go to playmolecule.org, you can try these tools yourself, and they are made freely available for you to use them
And
if you're interested in using them.
while this is computing, so maybe after this you already design your you may design some ligands that you want to try and you want to that are similar to the other ones, so basically a congeneric series of ligands, and then you upload them into this tool delta, okay, which is specific. It's relative binding affinity prediction, so it's specific for congeneric series. And here is the results examples that it gives. Okay, so here we have an example congeneric series of ligands, all right, and you can see that it gives you affinity values, but keep in mind though that these are obtained based on relative affinity prediction. So you must already have some information on the congeneric series. So usually maybe you have a series of, here we have, yeah, for example, here's information. So number of molecules that you input for the training, here in this example are 35, and you want to test 7 new molecules. So instead of doing experiments or doing expensive MD simulations, you just put it here, and in matters of seconds you get predictions on how good are these ligands going to be. I mean, is this modification that I did here going to improve the affinity, or is it going to make it worse? How much good is it going to be? How much worse is it going to be? And you just start playing with it, and it gives you useful information. And then, I mean, this is not exclusive, so maybe after you see this, okay, then maybe I'll just test 4 of these into experimental results, and so on. So here, the previous job we had computed to give you an example on how this LeadRing works. So yeah, here is the input molecule, okay, and here's the smiles, which is basically a text representation of the molecule, which is actually what the network outputs, okay. And here are examples of molecules that you could test that are similar to that one. and also based on its shape and with similar properties but different. So yeah, there's more different structures, like this one is highly more different. There's more weird ones, but there's also more simpler ones that maybe you haven't accounted for, and basically gives ideas on how you might design new molecules, on how you can add or not, and with molecules that make chemical sense and are not just whatever output. All right, and yeah, maybe after you have selected these lead compounds and you have this congeneric series, and you see some candidates that have good affinity, but maybe you want to test, okay, I know this molecule binds into this target, but I don't know if this molecule is going to bind to other proteins in other pathways that are going to affect in the animal trials. So maybe I mean, the molecule does its function into binding to a particular protein, but it also binds to other proteins and it causes problems. Okay, so maybe we can input that into pathway map. Okay, and this is an example output of pathway map. So you basically here I inputted an SDF file with 4 ligands. Okay, so you can see that the square every row is one ligand, and every column it's a different pathway that has a significant correlation with some of the ligands. So for example, this this 4th ligand we have here, it has a strong correlation with this pathway. So signal transduction, signaling molecules, endocrine system, circulatory system, and substance dependence. I mean, you can later go inside the pathways that are defined. It's either KEGH, the KEGH pathway database, or the Reactome pathway database, and you can look into which proteins are in there. But this gives you information on how your molecule is going to affect a human organism. So this molecule, for example, it's probably going to bind into something inside the endocrine system or into the circulatory system, which you might not want to. Okay, or also you can. I mean, if you have a compound that maybe you don't know what it does or if it has any effect, you just input it here, and it gives you information. Oh, maybe this might be valuable for cancer or for neurodegenerative diseases and so on. So it basically gives you information on which pathways your molecule might affect. And yeah, I think that's it for the ML-based applications. I don't know if you want to show any ML-based.
Yeah, we don't have. Yeah, we can show a couple of them. There's not much time left, but we can do a couple of them. So, yeah, for MD applications, I think with the time that we have left, I'm going to focus a bit on for example, membrane builder. Okay. So here, as you see, the interface is very similar. So here, for example, for membrane builder, so to build a membrane, you can select the membrane composition that you're interested in. For example, I'm going to use, so this POP-C is a type of lipid, okay, cholesterol is also another type. So these are things that are found in membranes. You can have like mixtures of POP-C with POP-E, for example, an example that I'm going here. So this is the size of the membrane. So membrane is normally a bit bidimensional. So so basically you select just the X and the YZ, the X and Y axis, okay? Then you can put here some membrane name if you want. And then you can decide as well on the ratio that you want, okay? So for example, 60% POP-E, 40% POP-C, okay? In the lower layer, and then on the upper layer, for example, keep this thing, and then you have to submit a name of the membrane, example, okay? And then you just submit, isn't it like this? Okay. Then it's going to compute. I think this one is relatively fast. But basically what it does is that it takes all the inputs that you had, and it basically generates a membrane that is going to be ready for you to then carry on with the rest of the process of building your system. So this one in particular, let's wait a bit while it finishes. While it finishes, I can also show protein prepare as well. Okay. Can I close this one?
Yeah, sure.
Just
close everything.
Okay. So you see that membrane builder is already finished. Okay. So you can see here that it has built a membrane based on the composition that you have selected. Okay. So maybe this may not be obvious for you, but basically, and actually the resolution also doesn't help, but basically the center of the membrane is here, where you have the upper and the bottom leaflet, and then you have basically the surface of the membrane. It's here, which is solvated with some water molecules, okay? And then this membrane can then be passed to the system builder together with the protein and with the ligand, so that it can build your system. Okay. Another example is the protein prepare. Okay, we're going to use the same example that Adrià was showing before through PTB. Okay, so again the same protein that we're showing. So you select, for example, pH 7.4. You say that you want, for example, to remove the water, that it already comes with the crystal structure. Okay, select the chain and then you just submit. Okay, I think this one is quite fast as well. And then other interesting things that I can show you. I think I will show you so another part, parameterize, for example.
Not much time, so.
Yeah, there's not much time, so maybe. Okay, so you see here that it shows a bit for example, it shows the residues with pKa close to 7.4. These ones are the ones that may be doubtful in terms of protonation state, which one you're going to choose. You have as well the protonation table, okay, where you can download an Excel as well of the protonation table. Wait, I don't know where does it start. Okay, so you have the residue name, the res ID, what is the chain, what is the pKa, okay, what's the protonation that it shows, okay. You also have the protonation diagram as well, the one that I was showing you before, okay, where you can see the pH that you've selected is here, and where sits the pKa of the residues of the protein, okay. This allows you to then download the current PDB, which is the current prepared protein with the current protonation states, or reassign some of the protonation states if you're not happy with them by selecting the different residues, okay, if you want them or not, you know, protonated. This one should be showing. Okay, this one is showing. So you can do some selections here, okay? If you want them protonated or you don't want them protonated. So what time is it?
Yeah, it's 6.
Okay. So we finish here. You can also, as I said, you can use this tool. You have it available on PlayMolecule.com, and if you have any questions, you can also email us if you want.
Email us, our email is here.
And we'll make as well the presentation available for it to be sent to you.
And yeah,
okay.
That's it. Thank you for your attention.
Thank you.
Thank you.